Welcome viewers. I am Professor S. Jeevan Chandra from Chaitanya Deemed University. I would like to extend the previous class, that is cell signaling, and today I would like to give a brief note on diversity of signal molecules and receptors. As like human beings, cells also have a language that is called biological language. The biological language is conveyed by means of different chemical systems. The chemical systems are called ligand molecules. The ligand molecules will searching for its own receptors. They are called receptors, receivers. Now they present on cell membrane, will receive the signals, and they recruit several biological functions inside. The receptors are a proteinaceous molecules which receive the ligand, can be of two origins: external, exterior location. They may present in interior location or intracellular receptors. There are biologically four types of receptors are there. They are called ligand interaction to the cell surface receptors will be of different types. They are ion gated channel linked receptors, G protein linked receptors, and enzyme linked receptors, and ligand gated cell receptors also there. Four classes of receptors are there in cell. Coming to how ions will be effectively convey the information, those receptors which are responded to ions, they are called ion linked receptors or ion channel linked receptors. Sodium channel, potassium channels, like that. When ions will searching for their receptors, upon binding, the signal molecules will induce a conformational change in receptor. Now, receptor will be open up, ions will be allowed from extracellular site to cytosol. In second way of proteins, example, some ligand molecules, signal molecules, will interacting with a 7 transmembrane protein. In a membrane, 7 domains are there, green color one. The ligand will going to bind to receptor, and it changes the cytoplasmic domain. The cytoplasmic domain will activate a yellow color protein called G protein. G protein going to interacting with the effector enzyme. They are called G protein coupled G protein linked receptors. Another type of enzyme receptors are there. That means receptor may recruit the enzyme activity, or receptor itself a enzyme. For example, enzyme linked receptors, tyrosine kinase receptors. Example. These receptors are monomeric in nature initially, but they require dimerization of ligand as well as the receptors. Now, inactive cytosolic domain and will be activated by signal molecule in the form of dimerization of the ligand, dimerization of receptor takes place, and cytosolic site is getting activated. Now it catalytically activate the down regulation of the molecules. For example, two different types of receptors may hold single molecule together, and they may activate the another enzyme system. Now activated enzyme system could be down regulating the cell. Coming to first class of G protein linked receptors, most of them are more 100 variety of G protein coupled receptor have discovered so far. They are found on cell surface. They contains 7 transmembrane proteins. In a membrane domain, there are 7 transmembrane proteins are there. Example, dark green colored zigzag structures found in the membrane. They are called 7 transmembrane membrane. And the extracellular space contains a receptor which is interacting with a ligand, and a cytoplasmic domain will going to be changing upon binding. Now what happens, it requires three important activities. First, ligand should be bind to its receptor, and then it activates the G protein subunit. And G protein subunit can be activated by hydrolysis of bound GTP from GDP to GTP. Now it's going to activate the down regulation of the signals. See here, what happens? Receptor is there, green color one, and inactive G protein always with GDP, guanine diphosphate. When GDP is there, it is inactive in nature. GDP should be replaced with GTP, and now activate the protein. How it is going to be activated? A signal molecule will come and resides on the extracellular space of the receptor. Now down regulating the activation of the signal. Now activated G protein will replace the GDP with GTP. As a GTP is located perfectly onto G protein, now it delivers the alpha, beta, gamma. Now beta, gamma will be kicked off from the complex. Now the activated beta gamma will down regulating the activation of beta gamma complex in cytoplasm will leads a different pathways. So activation of a target protein by alpha subunit also recruits different types of proteins. Now the dark brown color protein is called effector protein. The activated alpha protein going to interact with the effector molecule. Now effector molecule will hydrolyze the alpha subunit and cause dissociation from the target protein. Now target protein going to be activated. So alpha subunit will acting as a switch for effector accumulation. Now if alpha subunit with the effector it is activated. See here, if alpha is not with the effector molecule, it will be inactivated. This will be controlled by hydrolysis of the GTP. If alpha molecule with GTP it is activated, if alpha molecule with GDP it is inactivated. So addition of GTP, GDP, phosphorylation will acting as like a switch molecule. G protein molecule requires several mechanisms. First, interaction of the G protein linked receptor by its own ligand and activate the membrane bound enzymes, then increase the second messengers. When effector molecule will be going to recruited, that activates another molecule called cyclic AMP. Cyclic AMP will acting as the signal transducer. Now adenyl cyclase ATP converted into cyclic AMP. This cyclic AMP will going to activate the phosphodiesterases and this seed cyclic AMP dependent protein kinases leads to activation of CAMP proteins, cyclic AMP proteins. Both will leads to rapid and slow response based on the signal modulation. For G protein coupled receptors, three main secondary messengers are there. One is cyclic AMP, second one is inositol triphosphate and diacyl glycerol, third one is calcium. So IP3, DAG, calcium both are control over by G proteins, so it is called signal convergence. That means IP3, DAG, calcium both are controlled by same type of mechanism. They control the same type of calcium activity, so it is called signal convergence. So coming to second way of activation, the uh, G protein also activate the phospholipase C, will activate the inositol triphosphate and diacyl glycerol. Inositol triphosphate interacting with endoplasmic reticulum and membrane, it interacting with colmodyl receptor of calcium. Now calcium channel will be open up. The cytoplasmic calcium will be accumulated into endoplasmic reticulum. The greater than if excess amount of calcium is present in our cell, it will leads to many effects. DAG, diacylglycerol is a calcium channel modulator which is activate the phosphokinase C system to inner surface of the membrane and activate the phosphokinase C. This will leads to release of calcium. Release of calcium leads to cell relaxation. Free calcium, it leads to many effects on biological system by binding to different proteins called calmodulin receptor. Calmodulin dependent protein kinase will leads to muscle relaxation. If excess amount of calcium present in endoplasmic reticulum, that going to exert the cellular activations. That means either cell contraction, cell relaxation, both are controlled by calcium. So excess amount of calcium may not be good for the cell. So what happens? The activated G protein going to control the calcium release systems. How? When activated alpha interacting with the effector molecule, now it is going to recruit the cyclic AMP. Many intracellular messenger molecules are going to widely diffused into target proteins, and they control the signal proteins in various parts of the cell. Such a way it is called signal amplification. But here is a single G protein, but how many signal molecules are there? Many number of signal molecules are recruited. This way it is called signal amplification. So even cyclic AMP also acting as a signal molecule. How this is going to be controlled? ATP controlled into converted into cyclic AMP by means of adenyl cyclase. Adenyl cyclase are activated by G protein. Now cyclic AMP will be converted into cyclic phosphodiesterase, will be converted into 5 amino sin monophosphate, and cyclic AMP converted to AMP. Now biological activity will be turned off. So cyclic AMP are acting as a turn on switch, turn off switch. If cyclic AMP is there, it is turn on. If AMP is there, it is turn off. So that is called self-regulation. Now coming to how calcium will be controlled here, what happens? The activated cyclic AMP molecule controls a phosphokinase A. The activated phosphokinase A now diffused into endoplasmic reticulum or either the nucleus. In the nucleus, it activating the particular DNA molecule interacting with the promoter molecule. Now activate the this activated pKa will transfer the phosphate group to green colored target protein called gene regulatory protein. Now this regulatory protein going to bind to its own particular DNA as like a iron and a magnet. Now this is going to searching for its promoter region, and it will activating the targeted genes. Now gene transcription takes place, so RNAs are controlled. Now RNAs are converted into translated into proteins. Protein will take over the biological function. This is about cyclic AMP. So cyclic AMP is going to alter the biological function. The intracellular signaling pathway will leads to accumulation of intracellular signals, and they're going to diffuse the two-way action. Either they alter the protein functions in the cytosol, either they alter the regulatory elements of DNA and RNA. They are targeting the gene transcription. Finally, the proteins from translation, they control the cytoplasmic machinery, that is called cellular function. So all cells are targeted by these signal molecules, and the cell behaves differently by different types of signal molecules. So study of signal molecules, their corresponding receptors will explain the details about how cell is controlled by different type of receptors. Now we can understand the cell behavior, cell cycle, cell signaling, cell differentiation, tumor genesis, as well as the cancer cell and cell death. Why some tumor cells are not obeying the tumor activity, not obeying the apoptosis, cell death. These all are deduced very easily. How the G protein will control the calcium channel by diacylglycerol by activation of phospholipase C system. Activated G protein will activate the phospholipase C. Phospholipase C will attack the lipids, membrane bound lipid, inositol triphosphate. Inositol triphosphate was split into inositol triphosphate and diacylglycerol. Inositol 4,5 triphosphate has a high affinity towards calcium channel protein called calcium channel protein. Now calcium channel protein will be receive the inositol triphosphate, and it will open up the channel. Now calcium will be released from inside to outside. From endoplasmic reticulum, calcium will be released outside. When calcium is released, the calcium going to activate the phosphokinase C. Phosphokinase C requires calcium. So now calcium is released into cytoplasm. Now phospholipase C will activate the inositol triphosphate. Inositol triphosphate open up the calcium channel. Calcium channel will remove the excess amount of calcium present in endoplasmic reticulum. Now calcium will be released into cytoplasm, and which will going to activate the phosphokinase C. So phosphokinase C also further activated by byproduct of inositol triphosphate, that is nothing but diacylglycerol. Diacylglycerol also directly activated by itself through and it connected to PKC. But until unless it interacting with the calcium, the PKC is going to be activated. That means before that diacylglycerol along with IP3 are found on the membrane, but when it is lost the IP3, it still remain to associated with membrane in the form of diacylglycerol. This lipid is inactive when it is interacting with the inactivated PKC, but PKC along the diacylglycerol could be activated through a controlled manner called calcium. So now this is called signal divergence. So secondary molecule IP3 DAG both are controlling calcium channel in a stringent manner. So it is called signal divergence.